You can see it now, right?
Yeah, now in full screen.
Yeah.
Okay.
That's good.
All right, let's start it. Morning, guys. Today I'm going to do a journal club sharing, and I want to share it at once, so we can talk about the questions and the things that are interesting after all the all my sharing. Can we do it like that?
Yes.
Yeah. So basically, this is the article that I'm going to share today. Cellular senescence and the senescence secretory phenotype. Therapeutic opportunities by Tamara in a aging research laboratory in California, US. And yeah, basically, let's do some introduction first. Aging is the largest risk factor of most of the chronic diseases. You can see in the picture, it related to a lot of chronic disease which leading to failure, diabetes or sarcopenia, etc. And this article suggests that some of the recent findings suggest actually aging is a modifiable risk factor, and this article basically reveal the mechanism of aging and show how they modify the fundamental aging mechanism to delay those age-related disease. And within this article, it contains quite a lot of stuff, but these 3 points are the main points for the article. Basically, it reveals the mechanism and a term of SASP, senescence associated secretory phenotype, and I'll explain the term later, and it explains how cellular senescence and SASP associate with chronic disease and fertility, and it also point out the therapeutic opportunity to target senescent cells and SASP. And so let me explain what is cell senescence first. Basically, it is an age-related dysfunction or a chronic steroid inflammation. And what they mean by steroid inflammation is basically lack of microorganism, and which basically dysfunction the immune system and leading to inflammation. And it points the research recently point out that cell senescence is an important driver for many age-related disease, as you can see. And increase of senescent cells will basically result on oncologic cancer like response and threatened lifespan. As we can see in the article, increase in the cellular senescence will basically increase for our as long as we're getting older. Start from middle age of life, basically causing disease, as I mentioned, oncological disease, cancer, of atherosclerosis, metabolic imbalance, diabetes, etc. And at the end, we can die. So and the term SASP, senescence associated secretory phenotype, basically it just a DNA damage response, and how our body got the DNA damage response is by a lot of factors, as we can see in the diagram. Basically, the first one we know that DNA is damaged, and the oncologic mutation, it got lots of reason. It might include the trigger pathway that we learned before, mTOR, P53, P16, these tumor suppressor, and which trigger cell cycle or cell repair translation. And once they are triggered, they will basically induce senescent cells to produce, and SASP will be basically we can follow the flow in the diagram. Once senescent cells produce, and basically those IL-6, IL-1, TGF, these kind of inflammatory marker will increase and basically causing SASP increase. Basically, these SASP induce the mammalian phenotypes in nearby cell. What it means by you're gonna deteriorate the cell. Once one of the cell is got inflamed, it will deteriorate the cell that nearby, and then causing a lot of cell getting older and getting senescence situation. And it's very dangerous in the later lifespan, as it's causing a lot of aging phenotype, like the disease that we just mentioned before. So as long as we are thinking cell senescence is like a real bad thing, but actually this article also point out that is not 100% a bad thing. In the early lifespan, is a very key element to promote body evolution, as it builds up immune system to fight against either internal or external factor, and making us not to die in the early stages. And also enhanced senescence growth is basically beneficial for against cancer, because cancer, we are not sure what really causes about cancer, but basically cell senescence will cause cancer and curing cancer at the same time. I know it's contradict, but that's how the things works. And so the last part of this article also point out that it got a lot of therapeutic opportunities to target senescence cell, and it suggests 3 types of therapeutic opportunities. Let's watch it from left to right. The first one basically is interfering the pathways that produce senescence cells, like maybe tumor suppressor p53 or p16, or the one that we know a lot, mTOR. But this kind of interfering pathway will causing a lot of danger, because it might promote cancer at the same time, as it is not only affecting senescent cells, but it also affecting normal metabolic regulation within the body, which is causing a lot of co-effect that we are not really like 100% sure is it able to do it. And the middle one, second one, is just eliminating those already formed senescent cells. But some of the senescent cells are linked to neurons, and if we just eliminate most of them. Sorry, let me drink of water first. So basically, if we just targeting or eliminating them, we're just gonna kill most of the cell within the body, and causing a lot of damaged cells, which is not a good thing, because we just lock out all the cells within the body, but the body couldn't function. As what I mentioned, senescent cells is maybe got some function, build up immune system in early lifespan. It just got dysfunction within the body. We couldn't knock it out. But and the final one is the most possible one, is to prevent the development of SASP and ameliorating its effect. Basically, is to knock down, to lower its consequences or its bad, like its harmful stuff in the later lifespan. It's the most ideal one, and it's the most popular because it won't trigger any pathway or growth threats that will causing cancer or kind of serious chronic disease within the body. And also it's kind of interesting one. Some of the like external factors like dieting or calories restriction is actually shown that it can delay cellular senescence on health lifespan. And it's only it just point out one article that related to calories restrictions, which I believe external factors we have to be further investigated in order to to show we got a good relationship between, let's say, calorie restriction and delayed on senescent cell. And so how we target senescent cells, the article just point out that we can use antibody-specific making compound to kill them separately, and or second one, creating small molecules to kill them separately, or we just use vaccine, making antibodies again and deliver it into body, nucleotide created, and kill them. But we have to concern it. This part I'm not really sure. I haven't really go for it 100%, but senescent cell is not divided. Basically, it's a drug resistant, and it will be problematic when encounter compounds that target different cancer cell or microbes. And I think what it means, it's gonna kill those cells which are beneficial to our human body. So at the same time, it's not only targeting the harmful cell. And as what we mentioned before, cell senescence is actually not a bad thing in our human body, and it just not either kill them or leave them alone. And it's not as necessary to do it in order to reach the clinical benefits. And then conclusion for my journal club sharing. DNA, basically what is cell senescence is just DNA damage response and chronic inflammation, and what it causes, SASP, senescence associated
secretory phenotype.
And then though it's quite bad at the late lifespan, but actually it's beneficial in the early one. It just depends on the number of cell senescence it had, and what is the life stage in our own human body. And it's also pointed out there a lot of insight. Potential drug treatment can basically be developed based on this theory, as long as we really we can refine the direct relationship between some of the pathways and cell senescence. So dying is a must, but can we delay it or do something else to do it? And the thing I just pull up from my mind is about, after reading this, is 3 questions. Is it really necessary to remove it, and can we delay it through external factors, diet, exercise, sleep? And back to our lab, can ubiquitin target senescent cells directly? Which we can do something to test it. I don't know. And that's all I have to share today. Thanks.
Thank you, Jimmy. Could you open your last slide again, please?
This one?
Your question? Just remove senescence.
I haven't really think about deeply or read any other article cells, but I just think, I don't know, it didn't ubiquitin does a lot of stuff, right? Can they do it?
But in the paper, did they mention about the mechanism of senescent cell, their normal mechanism?
So
Right.
basically, back to here.
That one, I think
The one
just
on top.
I think that one is just like a pathway of senescent cell, right? But I would like to know the mechanism of senescent cell, how they I would like to know we can remove it by ubiquitin or other pathway. You know what I mean, right?
I think I get what you mean. What you mean is why the cell will get senescence, right?
Yeah, and I want to know because the paper said senescence have two functions, right? The first is good for body and another is bad for body.
Yeah, in short, yeah.
Yeah, so you have to know homeostasis, I think, about this cell. How the body control this cell to be good for the body.
Well, I think after reading this article, I think cell senescence is a must, basically a regulatory process for our body to get our cell to get repair within our chromosome. The things that make up our DNA. Like chromosome is just like shaped like that, can see it, and then like at the end of it, it got some part of called telomere, and basically, why DNA will damage, because as long as our body is regulating those, it just got and a lot of oxygen to produce in our body, and then we have to basically the DNA will get damaged because of that, and a lot of high glucose fatty acid, which those are toxic cell, will also damage the DNA and leading to like chromosome damage, and then the thing that I just mentioned, telomere, and then they're just like doing self-dividing and doing the cell cycle and like basically making a new chromosome again. But as long as the chromosome will eventually like from telomere go through cell cycle and become a new chromosome again. But at the end of it, it's just getting shorter and shorter, and that's how the cell die eventually, because it can keep replicating, but if it's just too small or too short, it would just like the cell would eventually couldn't replicate and gone. And then, but our body need to like regulate, right? So like here, like in the youthful function, we have to grow up, right?
Yeah.
If the cell is not senescence, we would just stick in the young stage, which is not a good thing, right? And then but at the in the late lifespan, we just you know what I mean.
I know, but I think from the picture it shows when the body get weak. No, just the red graph. It shows higher of cellular senescence cause the body weak or disease, right? Yeah.
So what's your question?
No. I just wonder because you mentioned about ubiquitination, right?
Yeah.
I would like to know if you're interested in ubiquitination of this cell, you have to make sure the process to remove this cell.
Which we need to know the pathway.
Know the pathway of this, because the pathway that you told me before is just the pathway to secrete SASP, it's just pro-inflammation, right? It's just if the cytokine is secrete a lot and it's not balanced, it will affect to cause the disease.
But I just wonder, can our lab do it?
Now I'm not sure. How to say? Now I don't know. Yeah.
Yeah.
I think there's some kind of like struggling with some of the fundamentals of underlying this. So I did look at senescence for a little bit before, and senescence, essentially, the hallmark of it is cell cycle arrest. So the cells don't grow and divide. Essentially, cells that are too damaged to really replicate, the body wants to stop replicating before there's, damaged mutations or potential for it to grow undividedly and turn into a cancerous cell line. So that's kind of like what P53 is a key marker of that. Other pathways that have kind of been implicated in it is like mTOR as well. mTOR pretty much turns off in senescent cells because it's not growing, it's not proliferating. So, but
is what, sorry?
mTOR is pretty much like just turned off. There's inactivation of mTOR essentially.
Oh, really?
So the cells aren't growing or dividing. Another thing that you can see sometimes with senescent cells is that they're also like quite enlarged in comparison to a normal cell. They're almost like swollen. So I think the one problem that you have with this is like defining a senescent cell is very difficult because it's almost like an umbrella term. You will see like a different phenotype. That's this whole senescence associated secretory phenotype. You will see different phenotypes from different cell lines. So it's not just like clear if there's one pathway implicating it. Potentially you could look at, the role of the UPS and perhaps if there's any sort of like buildup of polyubiquitin conjugates, if there's, perhaps a lack of, protein clearance taking place as, proteotoxic stress as you see there is a hallmark. But I think you'd have to go into more detail about that, I guess.
Yeah.
Brett, will you mind to explain a bit more about the cell arrest that you just mentioned?
So you see like P53 and P16. Those proteins are typically they're typically associated with like cell cycle. So like P53 is often called non-pkjim because cancer is uncontrolled cell dividing. In senescence, that kind of turns off. So it's kind of stop the body. In theory, the function of senescence is, like you said, it's a regulatory factor or a regulatory mechanism to control cell dividing of certain cell lines. So in theory, it's to stop cells that are damaged or in whatever way replicating themselves, as they would have a negative function on the body. However, the buildup of senescence is also associated as a key marker of aging. So, if you build it up too much, it's essentially, it's like JC said, it's all about homeostasis. Like you have some for function, but too much and over accumulation causes problems. Because there's so many cells that are now damaged because senescent cells, they don't do anything. They don't really produce much. Like you see here that all that's really released is certain cytokines. They don't really have like general function, I guess.
You mean cell senescence?
Yeah, it's kind of like to stop. Essentially, like the cells, like they're kind of in a state of they can't grow, they can't divide.
Yeah.
They build up and they kind of impact subsequent, organ functions depending on where they are. And it's a key like hallmark of aging. However, like the senescence associated secretory phenotype can be very, very different. It can differ between if you were to induce it in cell, what sort of treatment would you use?
Yeah, you're right.
Yeah, so like if you were trying to study senescence, for example, I know that YC wanted to, depending on what drug you would use, would probably impact the phenotype that you'd see, the SASP.
The SP.
The SASP, the senescence associated secretory phenotype, the cytokines and other things that they'd give off. So obviously, if the cells are in a dish and you then treat these cells and try and make them senescent, for example, using a cell cycle arrest, like a cancer drug, the secretory phenotype would be different depending on the drug that you'd use or the method that you'd use to cause senescence.
And the thing, yeah, and I think after you explain it, I understand the first point, that what we have to concern it now, because we couldn't really define which cell is senescence or if within the cell group.
Yeah, I think, because all the different, all the different cell lines from different organs are all going to have probably slightly different phenotypes.
Yeah.
I think that's the first point, is that if you're trying to then look at, well, in muscle, what sort of things are we looking at? What are we trying to then target? Because I have read some reviews and people were talking about myogenic progenitors in the past and other sorts of like stem cells and how they could potential to be beneficial for aging and also senescence as a topic.
Yeah, true.
I think my main sort of question is now, why did you choose this paper? Just out of curiosity.
YC gave me this paper instead, but yeah, I mean, it just, yeah, it matched, because if it is the area that YC interested, maybe our lab can really think about how you could like, suspicious association or relationship with it.
I think he I guess he interested in like aging mechanism. Right.
Yeah.
But when you you don't know the objective of YC, right?
Not yet. But we can build it up in the future, and especially ubiquitin is basically except what Brad is doing now, inflammatory origama, and other than that, it's just basically very heavy on skeletal muscle. And then, yeah, I mean, it just got a lot of unknown. Yeah, But it's still an interesting article. I mean, I really enjoyed reading it. Yeah. It got a lot of
Because I found I think last year I found the paper they mentioned about the experiment to study in senescent cell. Yeah, I think maybe that one will be useful for you.
Really? Can you send it to me?
I have to search again, because you try to study about this, right? But you don't know in our lab can do it or not.
What, study senescence?
It's about anti-aging. Yeah.
I mean, YC is trying to develop certain ways to study senescence.
Oh, I'm not sure. I just guessed.
Okay. Yeah. No. So he Rahul, JC.
What?
Rahul from Cambridge.
I don't know. Oh, that's contact. Or
yeah. So the guy who runs the lab down in Cambridge, Rahul, him and YC spoke, and he gave YC some protocols on how to measure senescence, at least to check if your cells are senescent.
Oh my god, interesting one. I want to do it.
So yeah, the one of the things is like trying to get. I think the drug that YC is trying to get in is quite difficult to get in, at least.
I got it. Okay.
So he wants to essentially, like, I mean, I can share my screen so I can show you some of the stuff that he sent me to look through.
Let me stop sharing it now. Yeah.
Okay. So this was quite a good one. This paper explains kind of a lot of the current protocols to look at senescence. As you can see, there's loads of different ways to do it. So you can use radiation. You can use doxorubicin, which is a cancer drug. You can use RAS, mitochondrial dysfunction. I think drugs used in like HIV and other things. So kind of the thing is you have so many different ways to kind of try and turn a cell senescent, because our main sort of thing with senescent is a cell cycle arrest. So you can see the cell kind of gets like big and loses its shape. That's kind of a sort of thing, and different ways to like look at it. So there's different types of staining, looking at gene expression, facts and other things. And then the other thing that you can use, is what other people are interested in, is the use of this media. So turning your cells senescent and then taking the media and treating healthy control cells with that and seeing the effect. So yeah,
I'm sorry. How to make the cell to be senescent
for
the serum media? Yeah.
Oh, for the serum media, so you treat with one of these drugs, whatever thing. So RAS overexpression, doxorubicin, that's what YC was thinking about using. In the medical school, they can use radiation. So you treat these cells with that, and then you can turn it senescent. And then you take the media, essentially from the cell you've turned senescent, and take it, and then use a conditioned media experiment. But I think the main thing YC wanted to do initially was looking at this doxorubicin, turn cell senescent, and then EDU staining to kind of show DNA damage.
Yeah, but Bradley, for the protocol 2, they will take only the supernatant, right? Not harvest the cell.
Yeah, just the supernatant. Yeah.
Why they did?
Because they want to see. So, do you remember Jimmy was talking about the senescent associated secretory phenotype?
Oh yeah,
i.e. what the cells are secreting outwards. They want to see what the cells have secreted and how that can interact. That media then can interact with other cells. How does it then turn these cells senescent? What sort of, what effect does it have on them? So it can sort of show how cells in one area can kind of like spread and become senescent. I guess we're trying to look at how the surrounding microenvironment of a senescent cell can be affected.
Yeah.
Would you mind to speak again? How? What? Wait, I didn't catch it.
Oh, that's fine. So how you were talking about the senescence associated secretory phenotype. So one of the common things that people see come off of cells is interleukin-6. How cytokines like that are released from senescent cells out into the media from a senescent cell, how that media then interacts with other cells. So, for example, you then put that media into a new dish of like healthy cells and seeing, well, does it turn them senescent? How does it affect them? Or how does it even like crosstalk between tissues?
Between tissues?
Yeah, different cell, right?
Yeah. For example, like in typical microenvironment, you're not just going to have muscle tissue. If your muscle tissue is close to like connective tissue on the outside, or how does immune cells that would then come into the muscle or be in the bloodstream, how would these then interact? You can sort of look at different sort of experiments. How does your senescent media interact with other cells? Depending on what you find. I don't know too much about that. I just know that's the kind of the point of the conditioned media experiment, and that from work they know that senescent cells kind of can turn other cells almost senescent. I think the main thing that YC is looking at now is basic protocol 1 and then B, the support protocol.
A and B.
Yeah, because Martin from the lab next door had some protein markers to look at senescence, for example, P53 and other things like P16 as well.
You mean we plot them and we can know the markers?
Yeah. So, well, I mean, they're cell cycle markers, and the main thing about senescence is cell cycle arrest, so that the cells aren't dividing. So if you show that the cell cycle marker, that it's basically not firing, that there's no cell cycle going on, that's kind of like a identification that they're senescent. The same with the EDU staining. If you're trying to look for DNA, when you put the when you like incubate the cells with EDU, you should theoretically see no color light up in your senescent cells because there's no new DNA being formed because the cells aren't dividing.
Yes.
However, then when you have a control cell next to that, it should be lit up green because they're comfortably dividing. The dye's being incubated into, well, sorry, it's binding to the new DNA that's been synthesized. So the first part is to make cells senescent. And then once you know that you've done that correctly, and that's sort of like your control steps, you can then go on further to look at different pathways and different treatments within senescent cells. Or, you can even feed back and look at how your different treatments might induce sort of senescent phenotypes almost.
Yeah.
Interesting.
Thank you.
Anyways.
So
Thanks, Brett.
Right.
That's cool. Can you send me this article a bit?
Yeah, sure. I mean,
but like, what is the cell that was he looking for?
I think he wants to do it on C2C12. So the protocol he got given, this is the protocol by Rahul. So Rahul did it. So this is Judith Campisi. She was actually one of the authors in the paper that Jimmy used. So she's also the one that did that current protocol one. She's quite important for senescence. But this is the doxorubicin-induced senescence. So with this, you have to be kind of careful with what cell lines you use. So they used fibroblasts.
For early stage one, though.
Yeah, IMR 90 fibroblasts. So what they do is they seed them to the T75 flasks, and they then they dilute doxorubicin. And essentially, the protocol looks like this, where they seed their senescent flasks. Once they're set, they then add doxorubicin for 24 hours, and then they replace the media, and then keep replacing the media. And then they have 3 different cell lines. They have the healthy control that they do nothing to; they just change the media and let it grow. They have starvation, which we call quiescent. That will reduce cell proliferation because you're not feeding it. If you don't feed the cells, obviously they're not going to grow and divide as much. And then if they're senescent, they shouldn't divide at all. So that way you kind of have 3 different ways to check if your staining is working. Because you'll have one cell with no EDU staining lighting up. There'll be no sort of like green fluorescence given off when you image it. You'll have one that has a little bit that's been starved, and then you'll have your dish that should just be lit up that is healthy and dividing. So he ultimately, I think he wants to do it in C2C12. You just have to be careful because you can't do it in cancer cell lines or epithelial cell lines.
Yeah, because eventually these will leading to cancer as well. Yeah, these cell lines.
I think it's just that the phenotype doesn't stay too long on them. I'm not too sure.
Not sure. Not me too.
I mean, I haven't really tried it, and then obviously to try with C2C12, you might run into other problems. It'd be nice to possibly have like some fibroblasts as a control, but I don't know if Martin has any still, because then you can check. Well, if it works in the fibroblasts and you've not followed all the steps, you know that it's working correctly, or if it's a teething issue using C2C12.
I think why they use fibroblasts instead, because just very early stage for the cell, and you know what I mean, like beneficial stuff in the early stage.
Yeah, but this is about what I know from when I was looking at this stuff before.
I need to steal all the articles from the Senescence cells and read them all.
Yeah, I can send them over if you want.
Yeah, thanks, bro.
Nice.
Yeah, I think that's what I can like understand from the article, and I try to make it short and share it, and no,
it was good. It was a good article.
Thanks.
Obviously, I'd say like if you're interested in your UPS and senescence, then I definitely look for what's out there, because it's quite an integral pathway. So I'm sure you can see some changes.
But I think not really. I mean, but this article is old. Not really a lot of people study it yet.
No, but it's fine. I mean, like I just mean just like, perhaps you could do a little bit of like general research, see if there's any papers out there, because I don't know of any papers looking at ubiquitin proteasome system. I think most of it focuses around cell growth markers.
Yeah, but I bet they must be related because ubiquitin regulates cell cycle as well, and cell cycle basically like related to cell senescence.
Yeah, and you
At
see
least
lots of
association.
But for my understanding now, cell senescence cannot die easily, right? Because they cannot go through apoptosis.
Yeah,
right?
I think they kind of
Really,
the idea that I have, I'm not sure if it's fully correct, is that once they become senescent, nothing's kind of really happening to them. They're just secreting cytokines and secreting their senescent phenotype. They're not really growing, dividing, or dying. They're just kind of in a state of nothingness, I guess.
Yeah, they cannot reverse or
But can they be cleaved or
what? Can they be cleared?
Yeah.
Potentially through certain mechanisms like identifying an antibody.
But these are external factors, right? Drugs or vaccines.
Well, I mean, they can be turned. Cells can be turned senescent by drugs. I don't know how things can look at like cell clearance from drugs. I don't really know what mechanisms they look at for sort of cell clearance, or how it'd go through, if it can really go through apoptosis. I don't really think it does.
It didn't go through what?
It doesn't really have cell-induced death. I don't know if immune cells can clear them. I doubt it, because of for immune cell to be able to clear a cell, it kind of needs a signal for it to an antigen for it to respond to, and if a cell's not essentially doing anything, it has no sort of growth, proliferation, or firing, then it's very unlikely that it's going to give off antigen signals. However, I haven't really looked at that. That could be something of interest.
I think it's the macrophage is going to do it, right? But if senescent cells stay in the body, just will causing more detrimental effect, Yeah, and you're gonna deteriorate the cell nearby and causing just cancer is already dangerous, right?
I think the main thing that is the problem with senescence is a loss of cell or organ function. That's why it's related to all the different diseases like neurodegenerative, like sarcopenia, because it's essentially a loss of quality of the organ that the senescent cells are within. Less cells are able to actually do their function of the organ.
Very new areas still.
Yeah, but so interesting.
Yeah.
Right. Yeah. Because now just aging people, I'll say now we have a higher aging people, right?
Yeah.
Yeah. I mean, there's so many aging people.
Yeah.
I think obviously for our lab, it's worth to try and look at like senescence just specifically at muscle level, and things associated possibly with sarcopenia, and looking at the reduced muscle quality and sort of sarcopenia, sort of the infiltration of fat cells and sort of like fibrotic areas of the muscle, and how is senescence a factor sort of contributing to this? What does it kind of do? Because it's not something I've particularly read in depth on. I was looking at essentially the overview of what senescence is and how can we induce it, because I think that's what YC wanted to first do.
Is it what is next project about? Or
well, that was just the main thing, is the first to essentially get down a model for senescence, and then the next thing, I think one of his interests is looking at how pro-inflammatory cytokines could be really contributing to the effect of senescence? Could they be driving sort of aging processes and the reduction in like cellular protein quality?
Yeah, I searched on Google. I don't know the detail yet, but they mentioned like exercise culture, the build up of senescent cell. Yeah, and helping the immune system clear the cell. Yeah, and fight the molecular damage.
Really? You find an article about it?
No, I just searched on Google because I wonder about senescent cell, right? Because senescent cell, like Bradley talk, it's just like cannot growth. It just like a bed in the body, and it's harmful because they secrete a lot of cytokine to cause inflammation, and inflammation connect to many disease.
Right?
Yeah. And I found like they mentioned like autophagy can remove senescent cell, but different condition. Yeah, I just searched on Google. I didn't know about the deep detail.
Yeah.
That I wonder about how to remove senescent.
Okay, so I mean, going forwards, is like how this topic is essentially important. Is it something you particularly want to study, Jimmy, or is it just more you were curious to learn about it? Because I think right now, like we've finished and we're just kind of like trying to hypothesize and understand stuff. But then the next question is is it really relevant to what you're going to do going forward?
Potentially, yeah. This is the more interesting thing I find out now.
But I don't know. Do you?
We're just learning,
Yeah.
But
yeah, no, I think that's fair.
It would be good if you know like your project.
Yeah.
Because you can read and focus on this, and then you can design your experiment and discuss with YC.
Yeah, I think I'm extremely interested in this area now.
About aging, about senescent cell, right? Oh.
Yeah. And then I'll go and talk to him at some point.
But I don't think you can't really do anything with senescence yet.
Yeah, but I mean, I'm still learning Western, like building fundamentals. I'm chasing you, and I mean, I need some time. So yeah, but I will chase it. Yeah, to get back. Yeah, talk. Yeah, but I would try my best.
Yeah.
Interesting one.
Yeah. So yeah, thank you, Jimmy.
What time you will be in the lab today, JC?
I will go to school after this meeting.
Okay.
Yeah, and I feel like I think now you can manage your time and make sure you are not doing the experiment alone. Like make sure you come at the time that Sam and I stay in the lab, because when you are struggle, you can ask I or Sam. Yeah.
Okay.
Yeah, because now I think you are ready to do it.
Yeah, but I just help JC to do some Western and practice a bit.
Yeah. I will give you some sample for practice. Yeah.
Okay. Yeah, I will do.
Yeah.
Sounds great. Yeah. Is it what you plan to do this week?
For me? Yeah, run Western blot and cell culture.
Okay. But seems Sam is going to do CRISPR tomorrow.
CRISPR Cas9 tomorrow. What time?
I don't know yet, but I mean last week that's what he said. He's gonna meet YC today and talk about what time he's gonna do CRISPR today. On Tuesday, I think.
Tuesday, okay, because I would like to observe him as well.
Yeah. He sent us a protocol about CRISPR from a protocol from Dundee. Do you want it? I can like.
Yeah, sure. I can have a look.
Just interesting one, but yeah, Sammy just say easy, like doing a cell culture, like should it fulfill and then done. I don't know. Yeah, I send you soon. Yeah.
Fair. Who's presenting? Is this on next week?
Yes.
Is it me presenting?
Yes, your turn.
Okay.
And can you send the paper within Friday?
Yeah, sure.
Yeah, because I have time to read on weekend.
Yeah, I'll probably send it over quite soon. Actually, I already know the one I wanna.
Okay.
I've got a few lined up on the topic I've been doing about. I think it's probably good for me to properly read through them and their tests and how they do everything.
Yeah, sure.
You get article related to immunoproteasome?
Yeah. No, I'm presenting. I mean, it came out quite a few years ago, but I think the group it came out from, I do quite like the work they're doing. So.
Yeah.
Okay.
Maybe maybe you have to just try to explain a bit more like fundamental to me, and
if anything's like, if anything I talk too quick or whatever, because I talk quite quick, just say like, you're going too quick. I don't mind slowing down.
Yeah, sure.
Yeah, anyway, right, well, I think that's it for today. See you guys soon.
Yeah, thanks.
Take care.
And you're on the trip, right?
I'm just at home for a bit because I've just been so busy throughout the year, just taking time off.
Yeah. So can I confirm a bit about Journal Club? Because last week YC asked me, he would like to he suggests to do Journal Club in person. Yeah. But I told him, we discussed last time, we prefer to be on Zoom in during summer term, right?
Yeah, I think like I'm quite busy at the minute I have stuff going on here that I'm trying to sort out. Then I go on holiday, so I can't really do too much. But then once, like from July onwards, I'll be in Birmingham pretty much all the time. So
that's fine. Just make sure we will start in person in July.
Yeah.
Okay.
You mean until graduate, right? Or you're gonna stay somewhere?
So once everyone starts moving flat, I get back to Birmingham on June 30th. So obviously then, I've moved a lot of my stuff out now, but I'm going to be moving to my next accommodation. So I'll be busy that day. But from then onwards, I'm a lot less busy. I'm pretty much just going to focus all my time on whatever I'm going to be doing for my master's to try and get that sorted.
I'll stay here till 20th of July and then back in mid of September. So during that period, I'm gonna ignore that time zone and do the Journal Club directly. But yeah, I'm gonna pop up when it's Journal Club one, but have to do in Zoom.
Yeah. Okay. Yeah. So
Thanks, guys.
Thank you.
Nice. Thanks, everyone.
Yeah, thanks.
Bye.